way it went was I was looking for what to do with my life when I was 15, 16. And initially I wanted to become a physicist, but then I realized that in order to make real discoveries in physics, you either have to be quite lucky or you have to be working for a very long time. So, if you have like one regular lifetime, then you might not actually be able to make significant impact on the field. But the way I discovered longevity was actually through Aubrey de Grey's book. It's called Ending Aging. And apparently it had just been published like a few years before I was 16. And so there was this one summer when I discovered both the genetic engineering and synthetic biology and longevity biology. And what Aubrey de Grey says in his book is, look, aging is actually just an engineering problem. There's these eight major things that he lists that are going wrong in most bodies that if we could engineer, the body would live longer. And that was like a light bulb moment in the sense that, oh, for the first time ever, we understand this mysterious process, aging, and we now actually also have the tools to do this, but no one had really ever done this and actually done, engineering on longevity. So that's how it got started.
Very cool. I remember learning a bit about Aubrey de Grey as well, and the hallmarks of aging and so on. What were there any other scientists back then that are sort of famous these days that, David Sinclair comes to mind. At least I learned about them too at the same time. I don't know if he was like known at the time in the longevity field.
He was probably known, well, he was definitely known because he had started and then sold this company, Surgery
Mhm.
Pharmaceuticals, or Surgery Bio. I don't know which one it is. And that definitely put him to the forefront of the field, but he was nowhere near as well known as today. So it was really a very small field, and it was kind of a backwater, really. People weren't studying it very seriously, except for a few labs. And yeah,
Very cool. So how about now? Like what brought you to building Olden Labs?
yeah, actually, the experience from my PhD.
Okay.
So by the time I got to my PhD, I had already learned enough genetic engineering to know that I could engineer the actual circuitry of aging of for longevity if the tools were there. But in order to be able to do that in a living organism like you and I or an animal, you first have to be able to deliver the DNA into the body, or you have to engineer the animal from scratch. Right? But if you engineer the germline, then you can only do it once per generation, so it's very slow and it's not scalable. So in my PhD, I set out to build a whole body gene delivery system that would basically allow us to take an adult person and change all the cells genetically with DNA.
How does that one
And
work?
so it builds upon gene therapy. Now, actually, I thought that the field was already kind of close because people write that, oh, we can do systemic delivery and so on. But it turned out that it was not anywhere near close to that. So in gene therapy, you have a genetic disease normally that affects one or a few tissues only. And then people try to deliver DNA into these tissues to fix the genetic disorder. And so I wanted to do this at the scale of the whole body. And I was using the same methods that other people were using, just building upon them. And the most effective methods to this day, for sure, by far, is the adeno-associated virus vector or AAV vector. So that's what I started working on in my PhD in Harvard. So I was doing this PhD across two labs, longevity lab and the gene therapy lab. And I had actually developed the system to be good enough to be effective for what we needed. So we were able to deliver to at least 80% of the body mass in an adult, like old animal. And I was super happy. It's like, okay, this is working. This is such a big progress. And I used it then to test out different gene therapies, which was always the goal, to do genetic engineering of longevity. And that's when I really hit the wall. And the wall was just doing the tests on mice.
Hmm.
And that's because whenever you put a drug into a mouse, you have to obviously measure like what's the effect of the drug on the health of the animals. And with longevity, you have to do this on many animals. You have to use hundreds because the effect size aren't very large and there's a lot of variation. So for you to get statistical significance, you have to use many, many animals. And that meant that I was working with somewhere between like 400 and a few thousand animals. And in my experiments, I used probably at least 600 animals when I did the actual therapeutic testing.
Mhm.
And I would have to measure all these animals. And just to get the basic health data, it took me almost a year of just doing nothing but manually measuring these mice all day, every day. And the way this looks like is, look, if you want to know how strong a mouse is, then I'm not kidding right now. You take a mouse, you put it on a grid, like a metal grid. So you flip the grid over and then you just stand there and wait. And you measure the time how long the mouse takes to fall. And the mouse can take maybe a few minutes if it's really old, up to a few hours if it's young to fall. And then you have to repeat that for every mouse to then get the average. And then you have to repeat that twice to get the average of different days. And then you have to do this for every single mouse in a like 100 mouse cohort. So you spend just standing there with a grid, like you spend days just doing that. And that's the state of the art. So I realized that if we don't solve the animal testing issue, then there's no way for even just me personally as a scientist to scale all the tests that I wanted to do. And I figured out how to solve it, technically. Like I came up with these technologies in my mind, but I knew I wasn't an entrepreneur. Like I'd always planned to be a scientist. So I actually spent a year trying to find an entrepreneur to build Olden, basically to build it for me. And I spoke to over 1,000 people.
Wow.
And I found some really great people, but they were all building much bigger companies. Like they would never just start with something new. So I realized that I had to build it myself. But I was super lucky that I met my now first co-founder, Noah, who was an AI engineer. So he had been building an AI drug discovery company. He was very, very good at this. And what we realized together discussing this was actually the data analysis was going to be the biggest issue because the hardware you build once and you get it done, but then the data is going to keep on flowing. You have to continuously work on that to pull out different health metrics from what you see. And so we saw that this was going to be an issue, and he had also gotten burned in his company trying to do animal testing. So he had used this other, these are called contract research organizations, where if you want to do a study, you give that study to them, they will do it for you, they will give you the data back. He used someone in India to be, to that he thought was reliable. Turns out it wasn't. They just lost 6 months because this company didn't deliver. So that was his experience, and we realized this was a systemic issue, and that's how we started building this.
Why had nobody done this before? Why was this the state of the art?
Yes, that's a great question. I think there's a confluence of factors there. I think the first factor is that animal research, just like doing the research, is kind of dirty work. Like, it's very repetitive. Animals, they actually smell, because they pee and poo on you and whatnot. They bite. And so it's not really like an intellectually stimulating work.
Right.
And I think for that reason, the kinds of very technical people that, build sequencing systems and are doing, everything else, they just don't go into that field. That's one. So
Hmm.
there's like a lack of talent. But also, it's actually a really difficult problem to try to automate this because, you have a whole animal and you have to make sure that their health is going to be okay. And then they have all these complex behaviors. And even, I can go into this, but even doing the tracking itself is computationally intensive. It's, you know, like just like physics based without, if you don't use vision, it's very difficult to do accurately. So there's all these technical issues that you have to all solve to get a better system than a human is. So it was a large jump to try to come up with something that's better than a human. And many people had tried. There's many smart cages out there. It's just that they were all either way too limited or they were way too expensive. And so in my PhD, that's why I didn't use them. They weren't more useful than my own hands. So,
Hmm. So is that because I think I got a little bit lost in the second reason? Is it like processing so much
sure.
information or the actual handling of I don't know, the sensors that you need or the how you're feeding the animal or like, all these mechanical stuff you need to do? What's the hardest part?
Yes, I think. it's a combination of difficulties. So maybe I can tell you what was the state of the art and then what we built, and that will give you an example. So the state of the art was this cage. I can tell you the name of the vendor. Maybe it doesn't matter, but it's this cage where they have a bunch of mechanical sensors inside the cage itself. So there's a weight sensor, there's a food sensor, there's a like water drinking sensor, there's a temperature sensor, and then there's like a basic movement sensor that tracks the animal's movement through near infrared. And with this kind of mechanical setup, it's kind of easy to set this up. The technology is not very difficult, and it's easy to track one mouse at a time. But the issue with that is that it becomes very expensive very quickly because a just a building the mechanical things, everything has to be made out of metal and every part costs. B, you have to clean the whole system every week, every couple of weeks because animals pee and poo on it. It just like gets dirty, right? It's very laborious to do that. And C, and the biggest issue is you can only do one animal at a time. But mice and actually mice, rats, pretty much most animals are social. If you keep them alone, then they actually get depressed, they get stressed, they get cold because they're used to being together, right? And as a result, you can't really do these studies for longer than maybe like a week or 2 weeks. So for something like longevity, where you literally measure the animals for 2 years, there is just not possible to use them. And for whatever reason, they were very expensive. So there was like $30,000 per one cage to be able to do one animal at a time for like up to 1 week. This is
You
like
said
much, much more expensive to
$30,000?
do. $30,000 per cage, yes, for one of these kinds.
Can you say already how much yours is?
Yes, it's right now $1,000.
Okay. So
Yes,
then what have you done differently? How is your cage designed? The domes, right?
yes. Yeah, so what we did differently was to try to solve all three major problems, which is how do you keep animals together socially but still track them all individually and be able to pull out their health? That's one. How do you make it very like I would say very cheap. Like $1,000 isn't necessarily cheap, but it's accessible for most labs. How do you make it accessible? And C, and that's probably the biggest issue, how do you make it scalable in a sense that when you have an animal facility, and I can show you what they look like if you want,
Mhm.
uh, you have a bunch of racks that fill out this specialized room. And in these racks, you have these individual cages. And in most facilities, at least in the States, as far as I know, the entire space is taken up. So you basically have no more space left because everyone is doing their research, right? So space is a big premium. And these rooms have to be specialized because they have to have day-night cycling, they have to have like air filtration, they have to have power backups, and like you can't just, at least up until now, you can't just put them somewhere, right?
Yeah.
So whenever you have something that doesn't integrate into this infrastructure, you can't buy many of them because you just don't have anywhere to put them. So that was the third big issue, which is how can we make this compatible with this infrastructure? And what we came up with, and again, here I can just show you what it looks like if you want.
Yeah, feel free to share
But
your screen if you want.
the lid itself looks like this. So in a normal cage, you have a cage bottom, then you have a food holding area called food hopper, and then you have the cage lid. So what we did was we took out the regular cage lids and we quote unquote made them smart. So we put a computer in here and we put a camera and a microphone and Wi-Fi capabilities. So what this does, all that this does is that it records audio and near infrared video, then it compresses this and then it uploads the thing to the cloud. And most importantly, it interfaces with the rack. So let's see, where's this? So if this is your mouse rack, then it can go straight above the existing cages without modifying really many things. So you can kind of scale them. Now, what happens in the background is where the really difficult part comes in, because once these devices which are now low cost, gather the data, the audio and the video, and then other types of data we can add later on, you have to analyze this all audio and video.
Yeah.
And what you're getting is um, what you're getting is a feed like this. Right? So there's multiple animals in the cage. They all look the same. They all run around, and they all do their things. They all, sometimes like cross each other. They climb on top of each other. So it's, as a human, you can't keep track of them for at all. But for you to do scientific experiments, you need to know what animals what, because you inject them, you treat them differently, whatever. You just have to have this capacity. And as we found, no one had ever actually solved the problem of how you track multiple animals at the same time in the same cage without mixing them up. Never. There are some systems that can do this, like RFID, that can kind of hack, but they don't really give you this kind of data. So this was the problem that we fundamentally had to solve. And it turns out to be a really difficult computational problem because to do that, you have to put 24/7 video through neural nets, and that's just expensive. So when we first started, the first best tool that was available, it would actually cost us $1,000 per cage per month just to do the compute on this neural net detection. And it was only like 60, 70% accurate in terms of it would mix these mice up very quickly. So with Noah and with our team, we've now done almost 2 years of development, like very, very intense development. And now we've gotten this cost down to about $50 per month per cage that we can charge people. And it's a bit less for us. And we can keep on bringing this down. And we are somewhere between like 90 and 99% accurate, depending on like how many mice there are, how much they move around, and so on. And that's because we built our own AI system in-house. There's actually now more than 8 neural nets running here to analyze all this data. We have pulled down the costs like very heavily, and we've also come up with our own hardware to mark these animals.
How
And
many
this
mice can
hardware
you,
looks kind of simple.
oh,
Yeah.
how many mice can you monitor, like maximum?
Nice. So there's no hard max for us. It's
Okay.
just there are guidelines in terms of how many mice you can keep per cage in these normal cages, and that it's up to
Mm,
5.
makes
So
sense.
far we're doing up to 5. Yeah.
Yeah.
But the last piece of the puzzle, which also took us almost 2 years to solve, was how do you individually tag these animals? Because at the end of the day, if they're not tagged, yes, you can track them. You can track them even pretty accurately, but you have to pay a lot for this because the neural net has to be like, very complex and the data needs to be large, and it's just not cheap and not scalable. So we developed our own metal tag, this ear tag, and that turned out to be really difficult because mice they try to pull everything off. They can chew through everything basically. So plastic doesn't work. It has to be biocompatible. It has to be like super light so that it doesn't affect the animal. So mice are already identified by ear tags. It's just the ear tags are very different. So yes, just doing the metal work was really difficult. But now we have these tags, and essentially what happens is we have a few neural nets that identify the animals themselves, identify the head and the body, and then a few neural nets that identify the tag, and then a neural net that identifies the number on the tag. And when they all synchronize, then every now and then you can see that the mice can kind of go out of field of view or they can kind of cross each other. But as soon as the camera sees the tag, it corrects again. And that's how we can get this really accurate multi-animal data now, where we know exactly what the mouse is doing.
I see. How
Okay.
do humans, I suppose humans use some sort of tags usually as well?
So actually, this is very rarely done. Again, before we came along, there was not many people that were doing this. They would only test one mouse, maybe two mice at a time. Now, there's many ways that you can tag a mouse. Like, for example, you could draw like a line on their fur or on their tail or whatever. But they clean everything up. So when in like 2 to 4 days, it's pretty much gone. It's not stable. And that's why it was so difficult to come up with a system that would be compatible with that. But the good news is that now that we have it, this is like the foundation of all the system. Now that we have it, we can pull out a lot of different information about their health, like how much do they move, how much do they drink, how much do they eat, how much do they fight. It's basically like if you were being recorded by a camera 24/7, every single thing that you did, then at the end of that, you could you could basically quantize your behavior into its very atomic parts, right? And that's what we're trying to achieve. Um, Basically, what we're building is a first system that is this phenomics platform, where you have genomics, you sequence all the DNA, you have proteomics, you sequence all the proteins. Well, now we want to sequence all the health in this one black box where you just put the mouse in there, get the full health analysis data, and you don't need to do these one-off experiments. So that's the goal. Obviously, we're not there yet, but I think we have broken the backbone on this problem.
I see. I saw on your website that you also have, around 60 something biomarkers that you currently measure. Like, what are some other things that you can, like, yeah, possibly features that researchers look for the most?
Yeah, so we're not at 60 right now. We are right now giving out 20,
Hmm.
and there's another 10 to 15 internally that we're working with. But so that depends on what kind of, first off, it depends on what kind of hardware you have on the device
Hmm.
and what you're measuring. But there's a lot of things that you can measure off of just the camera and the audio itself. And it's what I mentioned, activity levels, how much they rear, how much they groom each other, how much they fight, how much they mate, how much they eat, how much they drink, how do they build their nests, where do they spend their time, how, like, who is the socially dominant animal that you can all kind of pull out from this. And so most of the things where you're interested about just the health and behavior, we can read out from here because of course behavior is like the summary of the health. Also things like breathing rate and body weight, you can kind of do from the camera. But that's a place where we would start putting in extra hardware. So of course you can put in more hardware in the smart cage itself, the smart lid, or you can put hardware into the cage where now the camera can read these things out. So things like temperature, body weight, also how much they urinate, or like what's the what's the humidity in the cage, how much they vocalize, like how much they speak, what noises do they make, and pretty much everything that you can measure non-invasively. At the end of the day, we can measure. Also things like, I suppose, cognitive testing. So if you put certain objects in there, you can track how they interact, you can track how well and how fast they learn, and yeah, that pretty much everything that you do non-invasively we can measure theoretically.
Pretty impressive. Um, do you have any patents for this? I imagine either for the case itself or the algorithms.
Yeah, we actually have quite a few. I think now we own 6 patents on these smart cages, the smart lids, and various aspects of the system. And then on our algorithms, we haven't actually patented anything. And that's because when you patent, you have to make it public.
All right.
So all the algorithms are actually just kept internally, they're kept trade secret. And honestly, they're developing so fast that it's almost pointless to try to patent. We're getting better all the time.
Makes sense. That's amazing. I love how also, it's and you mentioned nobody in other fields maybe wanted to build this company or be the founder or and I think that it might be, you might be the best person to build this, right? You've been there, you feel the struggle of doing these sorts of incredibly repetitive and manual things. And you have like this major objective, which is, I suppose, something like accelerating scientific research. Tell me more about that, like the mission you see in this, how you see this company in like 5 or 10 years.
Yeah, for sure. First, I just want to, on the record, like make it clear that we were definitely not the first ones to try to build this. There
For
was
sure.
at least one fairly well-known company before, and they actually tried to build something quite similar, but they might have been too early technically, where AI just wasn't very strong and whatnot. They were just quite expensive, and they were pretty able to do multiple animals, so they kind of went under. But there was
How
some good
early
work
was this?
done before that. So I think this previous company is called Biome. I think they started in 2013 or
Hmm.
2015. and I think they exited, I think 2022 or 2021. And it was just when I was starting to do my studies, these small studies at Harvard, and I would have wanted to use the tech, but they weren't selling it anymore, and it just was going to be too expensive anyway. Um, but yeah, you were asking where I see this in next to 5 to 10 years. Well, actually, this is like the very version one system that we are building right now. This smart lid. It's meant to be low cost, easily accessible, easily usable. What I really want to build in the long run is a kind of a cloud lab in the sense that there are these automated labs for the rest of biology. So this is just going to keep on looping. It's going to get it's going to get kind of
No,
uh,
it's good.
but so there's these automated labs in the rest of the biology. There's nothing like this for animal studies at all. So if you wanted to go out and outsource an animal study right now, like some of the ones that I did in my PhD, it's like 500,000. So it's like super expensive. And it's humans doing all the work. They get you the data back. And then like you have no visibility except for what they tell you is happening. So the initial idea was actually to build out an automated cloud lab for animal studies. And that would have included the smart gauges that you just saw. And in fact, a more like an advanced version of these smart gauges with more sensors, many more sensors on them, and then robotics to do the animal care and handling. And that's where we want to be. I think in the next, maybe not in 5 years, but definitely in 10 years, I would like us to see us having many of these facilities around the world where robots do most of the care. And when you want to do your study, you just log on to a place like this, you specify what you want to do, all the data goes through these smart systems where you see it live, and it's just much lower cost so that people can access and can actually accelerate the studies. And that's all there because when you look at the longevity field right now, and again, I'm not trying to build a tech company here. Like I was literally trying to solve the problem of how do we do more better longevity studies because the most longevity study is, to a first approximation, the only way that we know anything about aging at all. Like if you can't make a mouse live longer, then you don't really know if you have an aging drug. But because it's so expensive, because it's so laborious and difficult, people don't use enough mice, so you don't get statistical significance. And almost no one ever replicates studies. And just people don't do enough of that. So basically, if we can make it cheap, fast, easy, accessible, then we want to drive forward the field with many people doing many more experiments.
Some people have also, talked about, oh, well, maybe for some things, we're not going to need animals anymore, organoids, and perhaps even in silico experiments might um, suffice. So I think in this case, since you've already mentioned like phenomics, it's also going to be like useful for behavioral studies or, yeah, what are some areas where you see this definitely we need mice for this and of course longevity as well, like you actually need to know how long the mouse lives, right? But yeah, curious to know like what are the like more niche or specific areas that you're targeting here.
Yeah. So you definitely need a whole organism to do toxicity testing if you really want to know the toxicity, right? To do behavior and neuroscience and to do longevity. Now, arguably, you also need a whole organism to do metabolism and therefore things like obesity and um, like pretty much anywhere where you have multi-system interactions, we can't really do this with chips or with organoids. Now, that's one aspect where you just need a lot of different organisms together. The other aspect is just the cost. So right now, if you try to do something with organoids or with chips, it's actually more expensive. Like you can have many more individual ones, but growing an organoid, keeping it sterile, um, keeping it consistent of a consistent size, like none of these things are actually solved. So the way that I see it is the organoid, and bear in mind again, I had all these options in my PhD. Like I wouldn't have been using mice if there was something better out there. And our lab was working with organoids, was working with modeling and everything. And organoids are a great additional screening tool. You know, when you have a lot of different molecules, you want to quickly know, like a biochemical assay or like an RNA-based assay, you can use an organoid because it allows you to test many things quickly. But then after organoid, you still have to take it to a full physiological system. I think organoids are maybe a better version of that or like a additional version of that, but they don't really replace animals in no way. And anyone that's worked with animals just understands this very well. But I think you mentioned modeling. I think modeling is the future of biology in a sense that biology is very complex, but it's not infinitely complex. It's like if you have a car, if you know enough about the system, you can eventually model it well enough. And I think the same is true for animal, like human bodies. But the thing with modeling, though, is that the level of complexity is so high that we can't just write them up into deterministic models. At least right now, we just don't have enough information. So, as far as I can tell, right now, the only feasible and maybe scalable model to get there is if you just have enough data and you keep on collecting the data, and eventually you can kind of train the AI to do that. It's kind of like what AlphaFold did.
Right.
But to
You're
do
also
that, you have to
collecting
have
that data.
exactly right. So to do that, you have to have high capacity to collect data. It has to be like, you know, digital. It has to be all the same format in the same place and everything. There's no way to do that without automation. So yeah, I hope that in the next 20, 30 years, we can be at least you know, one of the reasons why people are able to model the whole animal.
In that sense, what is your policy around data from from the domes you sell and the data that your customers collect?
Yeah, I mean, the customers own the data, of course. They, you know, they have to own it. There's no other way. What I do hope, though, is that we can either we or people themselves can just share enough of it, and or we can incentivize them to share by, I don't know, giving them discounts or whatever. You know, academics do want to publish their data. I think they want to do the right thing. It's just you need to make it easy for them and need to have a place where they can put it.
Right.
So
Are
I'm
you
kind of
also
optimistic.
becoming that place for them?
We don't know yet. Um, I think if no one else does, then yes, we will. And I think we will be well positioned to be that. But you know, this is now it's a different model, it's a different problem in some ways. So you first got to build the hardware, you first got to build the systems, and then you can think about, well, now what's next? Yeah.
Okay, super cool. What else? Anything else you want to share? Anything else you wish I would have asked you that I haven't yet?
Oh, well, I suppose I'm just interested in your take on longevity, which is many
Hmm.
people think that you maybe can't do a lot or that you shouldn't do a lot, or it's almost like a fool's errand. Like you can maybe optimize a bit, but you can't really make progress. I'm wondering, how do you see the field? Like, how do you see the space? What is your personal take on it?
I haven't been following the space quite closely. I think that scientists sometimes tend to underestimate the power of culture sometimes and timing in the markets as well, and how that
Mm.
also influences like the things that we fund, I would say. So the person I have been, even unwillingly following is Brian Johnson, right? And I think that the fact that influencers and just like thought leaders start to talk about it a lot more, like now longevity is a topic that I can talk about with my like non-scientist friends and family, right? And the fact that that is true, I think, will just incentivize more innovations. Like the economic incentives will be there, more of them, because of course they've been there always, I think, on the scientific end. And I think that's maybe where your question was going more. Um, the question that I wondered about previously as well is how are we going to get this to all of these to all of the cells, right? Like how are we, if the sort of fountain of youth is sort of gene therapy, then how do I make sure that I get it everywhere? And the brain honestly sounds like one of those biggest challenges, right? Like even if my body is perfectly working perfectly and like my organs are, age, epigenetic age 18, then what about all the memories? Does the brain have capacity to fill that in? These are questions that I don't know if people are currently giving answers to. So, yeah, I mean, now I'm coming at it from a very non-expert and just like curious person perspective.
Yeah, it's actually very interesting. Who knows what is the brain's capacity for memory? I mean, we know that people have lived to 120 and they probably still remember their childhoods. So maybe it's the case that you will remember the highlights, but the longer you live, the less of these highlights you remember. It's going to be an interesting thing to find out when people live longer. But I think on the first question, though, in terms of how can we get this to every cell, well, that was my PhD. And I think the answer there is we can definitely do it. Hitting the brain definitely was one of the hardest problems. But by now, it's like the hottest topic, the hottest problem in the gene therapy space. So there's at least 20, 30 labs and companies in the world, probably more, dedicated just solving this one problem of how you get AAV DNA into the brain.
Hmm.
And every single year, there's a paper that pushes the boundary. Now, just last year, there was one published where they created this humanized mouse and they were able to deliver very effectively into that mouse, even though that wasn't possible before by targeting the certain receptor. In the mouse, even in my own hands, I could hit about 98% of the cells in the brain.
Wow.
Now, the difficulty is finding the versions for humans for that. Humans are somewhat different, and it's more difficult to look for them because you can't do experiments as rapidly. But we can definitely do it. I think I have no question about that. It's going to be a matter of time. It's going to be a matter of economics, actually. But
Mm.
the way that I think about it is, at the end of the day, DNA is just like any other piece of information. You know, you can't just do it once. You have to keep on. It's like your computer. You don't get one OS upgrade and that's it. Like every now and then, as tech evolves, you get something new and new and new again. And fundamentally, I think that's the only way for us to really progress substantially in longevity because with DNA, it becomes stable, right? You make an improvement that's stable. Now you can make the next improvement and that's stable and so on.
Mm.
So you don't have to keep on taking these drugs. You don't have to keep on doing these interventions, which eventually you're going to be like Brian Johnson. You spend like 80% of your time just like living longer. So, so yeah, I think we're not there yet, but the technology is fundamentally possible and it's getting there very quickly. And I think that's how it's going to look like.
Hmm, interesting. You know what was interesting to me about one of the first times that I was learning about the Yamanaka factors was also like thinking about it, and by no means I even know like the basics of information theory, but like how, yeah, like does this align with all of the thermodynamic laws and, information theory laws of the universe? Like, first of all, like, yeah, just like if if we are remaining sort of in the same state and we are like putting energy into keeping ourselves young, let's say, then like is that going to be possible forever? Is that like something, is there anything in the universe that dictates yeah, otherwise? I don't know. It sounds
There isn't, and I think it is possible, but it's you are dealing with laws of probabilities, right? And it's actually interesting. That's why understanding evolution is so critical for biologists because without that, you're just flailing around. When you look at evolution of life on the planet, then you see that we humans are actually one of the longest lived species ever. There are much longer lived species, but there are not that many of them. There's maybe like a handful, maybe a few hundred. And in our entire evolutionary history, every ancestor has lived less. Right? So that means that basically evolution has built our longevity up to this state. Of course,
Hmm.
that means that we can also live longer. Now, if you were to live for, let's say, 10,000 years, so, then even something like, I don't know, a piano falling on you would become a substantial risk
Yeah,
because you've lived for so long. Then it's the
exactly.
same risk as just like a house collapsing eventually. But of course, there's different ways around that. Maybe you could build very good regeneration or whatever. The way that I think about it is information loss is only one of these things that happen to you as an organism. Eventually, it's the same thing. It's the second law of thermodynamics. That is, everything that's highly organized, there's many more ways to be disorganized, to break, than there is to be a whole and unorganized unit. So the reprogramming factor addresses one, I think, fairly narrow part of this, which is the epigenetic control of gene expression. Although, I mean, clearly this has some impact. But then there's other things like mutations, like actual physical damage, um, like extracellular matrix, like protein aggregations. The body is complex. It's like a car. You can't just
Yeah.
upgrade the computer of a car, right? So,
Well, you've got me some things to think about, and I think I might, yeah, I do want to return to, genetic therapies for humans and stuff like that, which was my initial passion rather than plants, to be honest. But yeah, thank you for this conversation, Michael. I really enjoyed it, and best of luck in what's to come for Olden Labs. Yeah.
yeah. Likewise, Sophia, right? Thank you.